welcome to the deep dive. Today we're jumping right into what feels like a massive intellectual shortcut, a new incredibly detailed map of the mouse brain.
It really is.
Our mission is to understand how researchers manage to reconstruct a single neuron connectome. That's the full wiring diagram, neuron by neuron.
And they did it by pulling together just a mountain of data. We're talking detailed 3D shapes of neurons, predictions of where synapses might be, and even genetics.
It sounds like the next frontier of neuroscience.
It absolutely is, because, historically when you tried to map the brain, you always faced this huge dilemma.
Right.
On one hand, you could use something like electron microscopy, EM, to get microscale precision.
Yes.
You could actually see the individual synapse.
Okay, super precise.
Super precise, but you're limited to a tiny, tiny volume, maybe a cubic millimeter. A grain of sand, basically.
So you could be incredibly precise, but only inside a closet.
Exactly. Or you could go the other route, use something like viral tracing. That gives you mesoscale coverage. The whole brain.
The whole city.
The whole city, but it's blurred. You lose that critical single-cell resolution because you're tracking whole populations of cells, not individuals.
So we've had this choice. See one street lamp perfectly, or see the entire city as a blurry smear from 30,000 feet.
And this work. This is the attempt to see the whole city and map every single street lamp to bridge that gap.
So how do they do it? What's the foundation?
Well, the whole thing is built on two really spectacular data sets. First, they have the full 3D morphologies of 1,877 neurons. They call this the F-1877 set.
Okay, F for full.
Right. And second, they have the dendritic morphologies, so just the input side of a much, much larger set, 18,370 neurons, the D18370 set.
Let's just pause on that for a second to give you some context.
Yeah.
Manually reconstructing a single neuron, tracing every tiny branch, they can take a grad student months.
Months of painstaking work.
And we're talking about almost 2,000 of them fully done, plus over 18,000 others partially mapped. The scale is just. It's monumental.
It is. And the crucial part, the thing that makes it all work, is that every one of these cells is precisely registered to a universal grid.
A common map.
A common map. The Allen Common Coordinate Framework, the CCF Atlas. This universal grid is what lets them analyze connections across the entire brain without any ambiguity. Everything lines up.
Okay, so let's get into the mechanics. How do you go from a 3D shape, all these complex branches, to an actual connection score? They didn't have EM for the whole brain, so they had to estimate.
Exactly, they had to estimate. And they did it using two distinct but complementary methods, which created what they call the arbor-net and the bouton-net.
Arbor-net, let's start there.
Okay, so method one, the arbor-net, is all about proximity. It's based on the classic idea that if two neurons are close enough to touch, they might connect.
A potential handshake.
A potential handshake. They quantify this with a term called a potential arbor contact, or PAC.
A PAC. How specific is that?
Well, they define a PACus as occurring whenever the shortest Euclidean distance between an axon arbor, the output wire, and the nearest dendritic morphology, the input wire, is smaller than 5 micrometers.
Okay, 5 micrometers. Why that exact distance? Is that arbitrary?
It's a great question, and no, it's not arbitrary at all. It's biologically grounded. That 5-micrometer window is roughly the combined length of the dendritic spine, which is the little nub that receives the signal.
The receiver.
And the axon bouton, which is the little swelling that sends the signal. So it's an educated guess for the space where a physical connection is actually possible.
Got it. So using that PAC rule, what did the arbor-net look like?
They found 12,926 potential connections among those F-1877 neurons. But what really jumped out, and you see this in the data immediately, is the sparsity.
How sparse are we talking?
Breathtakingly sparse. Only 0.37% of all possible pairings actually showed up as a potential connection. That's 99.63% sparsity.
Wow, so that just shatters that old idea, Peter's rule, that if two cells overlap, they'll probably connect.
It completely refutes the simplest version of it. Yeah, overlap does not equal connection. Far from it.
Okay, so that's method one. But method two, the bouton-net, sounds even stricter.
It is. The bouton-net is based on what they call predicted axonal boutons, or PBs. So instead of that general 5-millimeter proximity rule,
you handshake some.
Right. This method says a potential connection can only happen at one of the 2.57 million predicted boutons they identified along the axons.
So it's not just, is the axon near the dendrite? It's, is a predicted synaptic knob on the axon near the dendrite. It's looking for the specific hardware.
Precisely. It's much more restrictive. And because of that, the bouton-net has even fewer connections, only 4,909. That makes it even sparser at 99.86%.
It's fascinating. They build these two separate networks, one on proximity, one on predicted structure, and they're both incredibly sparse.
Yeah.
But the first big finding was that they weren't random, right?
Not at all. Both networks, completely independently, showed really strong modularity. When they ran a standard clustering algorithm, the arbor-net fell into 12 groups, A1 to A12.
And the bouton-net.
Same thing, 12 groups, B1 to B12. That repeatable grouping is a huge sign that there's a real non-random organization happening even at the single cell level.
Can you give us a concrete example? What does one of those modules look like in the brain?
Sure. If you look at the arbor-net clusters, for instance, clusters A7 and A12 are mostly made of neurons from the thalamus, specifically a region called the VPM. That's the gateway for touch and sensation from the face and body.
Okay, a sensory module.
Exactly. But then you look at clusters A8 and A11, and they're mainly from the striatum, the connectome, which is all about motor control. So you're already seeing the separation of function just based on the wiring.
Okay, so let's do a quick recap here. We've taken thousands of single neuron shapes, put them on a master grid, and then used two different rules, general proximity, the passels,
right,
and predicted synaptic structures, the PDs, to create two different but highly sparse and modular connection maps.
That's a perfect synthesis. The key takeaways so far are, first, the challenge, single-cell resolution across the whole brain. Second, arbor-net. That's based on pieces, the proximity model. Third, bouton-net, based on PDs, the predicted hardware model. And the first big insight, both networks are super sparse, but they independently organize into 12 non-random modules that map onto real anatomy.
So we have two different methods giving us two maps, which brings up the big question for you, the listener. Since these were built on different principles, one looser and one stricter, how consistent do you think they actually are? Do they point to the same connections?
That question of consistency, of cross-validation, is really the heart of the next step. I mean, without whole brain EM data for a gold standard check, they had to show that their predictions align with each other and with what we already know about the brain.
So how did they stack up, PVs versus PVs?
They correlated really strongly. Initially, the raw correlation between the distributions was R equals 0.65, which is good.
Okay, pretty good.
But here's where the massive data set flexes its muscles. When they recalculated the packs using that much larger D18370 dendritic set, the nearly 18,400 neurons, the correlation jumped significantly, all the way up to R equals 0.76.
Wow, that's a huge jump. So by just adding more of the surrounding cellular context, the model got over 10 percentage points better.
Exactly. It shows both methods are capturing a similar biological reality. And what's more, they looked at the actual spatial alignment. Nearly a quarter, 24.58% of all the predicted boutons were found within a super tight 1 micrometer distance of the pack.
So they're not just vaguely in the same region, they're right on top of each other.
Far too often for it to be random chance. It strongly suggests they're identifying the same high probability spots for connection.
Okay, so they're consistent with each other.
Yeah.
But how well do these models reproduce circuits we already know exist? The classic pathways for things like vision and movement.
They do it very well. Let's take the bouton-net modules as an example. Look at the primary visual area, VISP. Neurons there were found to get about 43% of their inputs from other neurons right there in VISP.
Okay, local connections.
And a similarly large chunk from the LGD, the dorsal part of the lateral geniculate complex in the thalamus.
That is a textbook visual pathway. The OGD to ESE circuit is fundamental for vision. The fact that a model built just on neuron shapes can reproduce that that's huge.
It is huge. Because it suggests the morphology itself, the shape and where the boutons are placed, contains enough information to predict these major functional highways.
Amazing.
And we see the same thing in non-cortical areas, like the connectome, the CP, and the striatum.
Tell us about that.
So CP neurons in the model get most of their input from motor cortex, but their outputs project to places like the substantia nigra or globus pallidus,
which perfectly matches the direct and indirect dopamine pathways, D1 and D2, that control movement.
It perfectly aligns.
Yeah.
A model built on shape alone correctly reflects this incredibly complex push-pull system for motor control.
It's just incredible. So the model is holding up against decades of established neuroscience. Now, to bridge their single-cell data with those bigger, blurrier mesoscale maps, they created something called the CUNet.
Yes, the CUNet. It's basically their merged best of connectome, combining the scores from both the arbor-net and the bouton-net.
To get the most robust possible connection score.
Exactly. And then they compared the CNet to two existing mesoscale maps. The Allen-net, which uses viral tracing, and the BRICSEC-net, which uses single-cell barcoding.
And what did they find?
Well, first, the connection strength correlated positively with the BRICSIC net at around R equals 0.26, but more importantly, they checked it against whole brain synaptome data, a map of synaptic proteins, and found a really solid correlation of R equals 0.49.
Now that is a critical piece of evidence.
Yeah.
It means their predicted connections don't just align with other shape-based models, they align pretty well with the actual biochemical reality of where synapses are.
Absolutely. Yeah. And maybe the most powerful validation metric came from the comparison to the AllenNet when they looked at the absence of connections.
What they predicted would not connect.
Right. 97.9% of the non-connections they identified were also non-connections in the trusted AllenNet.
That is an astonishingly high number.
Right.
So the ability to confidently say these two regions don't talk is almost perfect. That could save researchers so much time chasing false leads.
For sure. And finally, circling back to modularity, they linked these modules to function. For example, modules B6, B8, and B11 were all strongly linked to vision, full of cells from VISP and LGD.
That makes sense.
But they also found something more general. Functions related to motion and feeling seem to be everywhere, distributed across the whole network. The specific senses, smell, taste, hearing, they were confined to very specific, specialized modules.
Okay, let's recap this validation segment. We've confirmed the models are statistically consistent with each other, they reflect known anatomy like the visual pathway, and they match real synaptome data.
Yep. Let's bullet point it. First, the consistency. That PAC-PB correlation jumped to a strong R of 0.76. Second, the validation. It confirmed canonical pathways and aligned with the synaptome map at R equals 0.49. Third, the mesoscale match. The SEU net predicted non-connections with 97.9% accuracy against the AllenNet. And finally, the functional modules. They found specialized modules for senses like vision, while motion was more general.
So we've established the anatomy and the function lineup. But the brain isn't just a physical machine, it's built from a genetic blueprint. So beyond shape, what about our genes? Did they find a link there?
They did, and this is where the single-neuron data really shines. They looked at gene co-expression patterns from other big data sets to see if connected neurons were more genetically similar.
And the big finding was that their single-neuron SEU net correlated more strongly with gene co-expression than the old population-level AllenNet. Why does that single-cell resolution matter so much for genes?
It's all about filtering out the noise. When you look at populations of cells, you're averaging everything together. That includes all the housekeeping genes, the ones every cell needs just to stay alive.
So two totally unconnected brain regions will still share some genetic similarity just because they're made of living cells.
Precisely. And that creates this background noise, this false signal of similarity. So what they did was filter out all those ubiquitous genes and focus only on the top 10% of the most highly expressed specialized genes.
The ones that really define what kind of neuron it is.
Exactly. And when they did that, the mean gene co-expression score for connected neurons in their SEU net was 0.35.
Good.
That score is 52% higher than the score for the population-based AllenNet, which was only 0.23.
52%. That is a massive difference. It suggests the genetic link to wiring is much stronger than we thought, but it was just being washed out by the noise in older models.
That's exactly it. By getting down to single cells, they found a much cleaner signal, linking the genetic instructions to the physical wiring. It's a profound link between the blueprint and the building.
And they also looked at the structure of the network itself, right? The building blocks or motifs.
Right. They focused on triads, so groups of 3 neurons. And they found that both the arbor-net and the bouton-net had nonrandom patterns. Specifically, a certain type of triad called M9 was overabundant. It showed up way more often than you'd expect by chance.
And the fact that it was overabundant in both networks, which were built independently,
suggests it's a real fundamental building block that the brain uses. And the neurons making up these M9 triads were similar in both networks, mostly coming from key sensory and motor areas. It all just reinforces this idea of deep, non-random structure.
And finally, they found that the network properties were consistent with a scale-free network. For anyone listening, what does scale-free mean here?
Think of it like an airline map. A scale-free network has a few major hubs, like Chicago O'Hare or Atlanta, that are massively connected. Okay.
But most airports are small, regional ones with only a few connections. The brain seems to work the same way. A few neurons are massive hubs, while most are only lightly connected.
And this scale-free property had been seen before at the mesoscale, right?
It had. So the fact that this single neuron data shows the same global property means these tiny local connections are following the same deep organizing rules that govern the entire network.
What a monumental piece of work. It really feels like we've moved beyond that old, generalized, if they're close, they connect rule.
We have. We've moved to a much more realistic estimation by including the actual predicted hardware, the PBs. This is that rich granularity of the mouse brain network that we've never been able to quantify at this scale before.
And it provides a reliable, practical framework while we wait for the technology, like whole brain electron microscopy, to catch up and verify every single one of these connections. And that ability to predict non-connections with 97.9% accuracy and to tie it all so strongly to gene expression, it's just groundbreaking.
It is. Which brings us to our final provocative question for you to think about. If this work shows that single neuron connections correlate so much more strongly with our genes than we ever realized, what undiscovered genetic mechanisms do you think are waiting to be found once this kind of mapping becomes routine?
What are the specific genes that tell the brain exactly where to put those M9 triads? The blueprint is clearly there, and it's far more detailed than we ever thought.
A deep dive into the blueprint of the brain. Thank you for joining us.